First thing we have to do, all this stuff takes a while. What we're going to do is use IPA first. And I really like IPA. I would say IPA is probably the gold standard of all this bioinformatics software.
Move your screen.
Oh, geez. Yep. What makes IPA so special? It is a human-curated database, which basically has mined the entirety of the scientific literature. So every relationship, so the legend has it that there's this island off the coast of San Francisco, and they've just got about hundreds of postdocs chained to their computers, just going through the scientific literature, calculating all this. You know, basically this gene interacts with this gene. That's what makes this different. It's the human curation on it, that it makes it much more accurate. Has anybody here used David? Some people, yeah. David sucks. It really does. It's a glorified text miner, and there's no human involved. And the last time they updated it, they actually just updated it now, but before that it was like 2010. So this stuff, it's commercial. It gets updated all the time. That's why I really like it. But first, so what we're going to do is we're going to take the genes that we feel are different or playing a role in activated B-cell lymphoma. So this is the aggressive B-cell lymphoma. We're going to take that gene list that you just calculated and we're going to throw it into IPA and see what pops out. So here on your window, so everybody has IPA open. Oh, jeez. I get too excited, I guess. All right. So in your window here, new, hit new core analysis. Everybody see this window? New. So we clicked new and we hit this button here. We got core analysis. Go upload a new data set. On your desktop is a folder. It's going to be different on mine, but on your desktop is an AACR folder. And in it, this is where you downloaded your BAM files. What you want, I've been analyzing so many of your data sets that like I can't find mine anymore. ABC versus GCB SIG gene list, N748. That's what you want. Double click on that. Here is where we say, here's where we tell the software what we're putting in, just like we did for the SeqMonk. So in the second column, Bob was very kind enough. Thank you, Bob. Did Bob leave? He did. Thank you, Bob, in the hallway, for putting full change information. So now we know a direction, which way these genes go in the activated form. So in your second column, put observation 1. So those of you that have used David, do you put full change information in there? No. If your bioinformatics software doesn't care what direction your gene goes, it's bad software. Don't use it. Find something else. The direction of your gene is very, very important. The way it's ranked is very, very important. So go to your third column here. It says adjusted diff p-value. Here we're going to put, this is also observation 1. Your full change and p-value come off the same statistical test. Okay, they're both observation 1. A lot of people get that wrong. Now go all over here. You're going to see this column that says ID, and it'll have an ENSG00, some numbers. Make that your ID column. We could use our gene symbol, but in my experience, you get more hits when you use the ensemble ID. Okay? So I've selected that. Now, this is telling me how many of those genes it's recognizing. It's recognizing 0. So what I have to do is I have to go to the select identifier type and make sure I tell it that I'm using ensemble. So click on that ensemble thing. What's nice about Ingenuity is I can have a lot of different IDs, like different types in my list, and I can basically, here's what I do is sometimes I don't even know what the ID is. I just kind of do Russian roulette and do hit select all, and hopefully something pops up. But in this case, we know it's ensemble, so hit OK. No, you can leave it. It's fine. After that, your data set summary should say 713. Does everybody say that? Rock and roll. All right. So go over here and hit Save and Create Analysis. You can name it whatever you want. I've got a bunch of lists on here. I got to put it something else, so I'm going to put 9. You can choose your project. You put it in your training project, or you can just create a folder, AACR 2016. And again, you can put notes. Hey, I did this with, SeqMonk, and I use these kind of parameters.
Can you hold on one second? It's not showing B17.
What's up?
For whatever reason, the gene list here doesn't show, it still says 0, but everything else looks right.
Okay, you don't have, this is wrong ID.
Oh.
You want ensemble ID here.
Yeah. Okay.
Now how are we doing? 713.
Yeah.
Yeah, you're good.
Is this under core? Is it under core analysis?
What's that?
Is it under core analysis?
Core analysis. Yeah.
So I'm on here, but
Okay, now you're at the, now this is where we are. Okay.
And then you said to save and create.
So hit Save and Create. Now, what we have done, this first step was telling the software what kind of file we gave it. Let me close it. Let me shorten this window. A little glitch there. Here is what's cool about this. So basically, this is where we tell the software how I want to analyze it. I would recommend just leaving everything as the default, since you're novice users. But I want to let you know about one thing. Click on this tissues and cell lines. What this screen allows me to do is say, given a collection of genes, I only want to see those genes that are known to be expressed in a certain cell type. Why the heck would I want to do that? Anybody? Think about tumors. What's in tumors besides cancer? What's in tumors? Stromal cells. What else? Immune cell. It's a garbage can. You got epithelial. You've got all these different cell types. What if you're only concerned in, say, the activated TH cells? I can tell this software and kind of filter that mess and say, show me only those genes known to be expressed in T cells. And it'll do that. This is a very good thing to use. The more cell types you have in your sample, the messier your analysis will be. We are working on cell lines. Our data is very, very clean. We got very, very clean data, especially given the fact we got 700 and some genes at a false discovery rate of 5%. It's pretty good. Okay, leave everything like it is, hit Run Analysis. And you can name it whatever you want. All right, we got it in. We just sent this gene list to supercomputers in Silicon Valley, and what they're doing is they're taking their database and they're crunching our gene list with their database. Once they're done crunching it, they're going to send us back the results. Okay? All right. Now let's go to BaseSpace.